For Module 12, we're going to continue our discussion of lipoprotein metabolism and link lipoprotein metabolism with cholesterol synthesis and transport in the body. For this, you'll need to have read Chapter 34. So, again, we're going to focus on 3 different points here. First, we'll look at cholesterol. It comes from both the diet from animal sources and is synthesized in the body. The majority of cholesterol in your body is from endogenous synthesis. Most all tissues can synthesize cholesterol as it's used as a membrane sterol, although the liver is our primary organ for synthesizing cholesterol. Cholesterol also plays an important role as it's a precursor for the synthesis of our steroid hormones. Cholesterol, like we've seen with triacylglycerols, is transported via lipoproteins. Our dietary cholesterol is incorporated into chylomicrons, and cholesterol synthesized in the liver is packaged into VLDL particles. These VLDL particles, carrying our newly synthesized liver triglycerides and cholesterol, circulate and will mature into LDL or low-density lipoprotein particles. These particles are taken up by the cells via the LDL receptor, and in this way cells are delivered cholesterol. Finally, for cholesterol transport, we'll look at how cholesterol from the periphery is transported back to the liver, and that's via HDL particles, or high-density lipoprotein particles. We'll summarize this by looking at the regulation of cholesterol synthesis. This pathway, as I mentioned, is regulated on several levels, at the level of phosphorylation, transcription, as well as feedback inhibition. The statins are a class of drugs that are cholesterol-reducing drugs, and these drugs we'll see target the enzyme HMG-CoA reductase. So let's look at the structure of cholesterol. It's a ringed structure, shown here, with a hydroxyl group on the 3-carbon. This hydroxyl group allows the molecule to be amphipathic, meaning having both hydrophilic and hydrophobic side chains. As a result of this, cholesterol can insert itself into the membrane of the cell. It's a planar molecule, meaning it's flat, and can influence the fluidity of a cell membrane. Approximately 1/3 of the cholesterol in the body is in this free form. The other constituent of cholesterol is esterified, and it's esterified at this hydroxyl group. The addition of a long-chain fatty acid, such as linoleic acid, attached at this carbon via an ester linkage, keeps the cholesterol within the cell and out of the cell membrane. This is important when we look at the transport of cholesterol. Essentially, the esterification of cholesterol is a means of storing it within the cell. The overall synthesis of cholesterol to generate this cyclic structure requires a lot of energy. The energy is supplied in the form of NADPH. And through a series of reactions, acetyl-CoA is condensed to form our cyclized compound. So like many pathways that we've seen, synthesis of cholesterol starts with acetyl-CoA. And this acetyl-CoA can come from any number of pathways. It can come from glucose oxidation, beta oxidation of fatty acids, or from ketogenic amino acids. In this first step of cholesterol synthesis, we generate our intermediate mevalonate. So in the first step, we have the condensation of two acetyl-CoAs to generate aceto-acetyl-CoA. This aceto-acetyl-CoA will condense with yet another molecule of acetyl-CoA into a reaction to generate our HMG-CoA intermediate. And again, our enzyme here is HMG-CoA synthase. This pathway should look familiar from ketone synthesis. Although the pathway is similar, these reactions take place in the cytosol, whereas ketone synthesis takes place in the mitochondria. Consequently, these enzymes are different as they're separated into different compartments within the cell. The regulatory point of this pathway is here at the enzyme HMG-CoA reductase, and this is our committed step converting HMG-CoA into mevalonate. HMG-CoA reductase is a large enzyme containing 8 transmembrane domains, and it sits in the membrane of the endoplasmic reticulum. The energy required for this reaction come in the form of 2-NADPH. The rate of synthesis of HMG-CoA reductase is highly regulated. It's regulated at the transcriptional level and is controlled by the family of sterol regulatory elements. So cholesterol regulates the transcription of HMG-CoA reductase in the following manner. When cholesterol within the cell is high, so the regulation of HMG-CoA reductase via the sterol response element is controlled by the levels of cholesterol within the cell. When cholesterol levels are high, they bind this transcription factor complex that's in the endoplasmic reticulum membrane. This complex consists of 2 proteins, SCAP and SREBP. This stands for sterol response element binding protein, while SCAP is sterol response element binding protein cleavage activating protein. These transcription factors are part of the helix-loop-helix transcription factor family. As cholesterol levels in the cell decrease and cholesterol is freed from this complex, the transcription factor complex is moved to the Golgi membrane. Within the Golgi, there is a cleavage reaction that releases the DNA binding domain of these transcription factors. This process is facilitated by 2 separate proteases, and the DNA binding domain of the transcription factors translocates to the nucleus. Once within the nucleus, it will bind the regulatory element or sterol response element and will regulate gene transcription. It's through this manner that HMG-CoA reductase is increased when cholesterol levels are decreased within the cell. HMG-CoA reductase is also directly impacted by sterol concentrations. Under high levels of sterols within the cell, the HMG-CoA reductase protein is targeted for proteolysis and degradation. This decreases the amount of protein within the cell. Finally, HMG-CoA reductase is also controlled by the level of phosphorylation. Under high levels of insulin, a phosphatase is activated and dephosphorylates HMG-CoA reductase. The enzyme is active in its dephosphorylated form. High levels of AMP, or the presence of glucagon or sterols, will activate AMP activated protein kinase, which will then phosphorylate the HMG-CoA reductase. The phosphorylated form of the reductase is inactive. These three mechanisms, the level of transcription, the level of protein degradation, and the level of covalent modification, all ensure tight control over cholesterol synthesis. From our starting point of mevalonate now, there's a series of reactions that add 3 phosphate groups to mevalonate. One here, an additional group, and one here. The purpose of these phosphate transfers is to activate both carbon 5 here and carbon 3. In the next series of reactions, the phosphate group from carbon 3 is removed, as well as the carbonyl group, to generate a double bond. This intermediate is the first of 2 activated isoprenes that are required for cholesterol synthesis. This isopentenyl pyrophosphate can then be isomerized to the dimethylallyl pyrophosphate. These 5 carbon precursors will be our synthetic precursors for the synthesis of cholesterol. For the subsequent reactions to generate cholesterol, it involves the head-to-tail condensation of our 2 5-carbon precursors, dimethylallyl pyrophosphate and isopentenyl pyrophosphate. This initially generates our 10-carbon geranyl pyrophosphate intermediate. The addition of another isopentenyl pyrophosphate group generates our 15-carbon intermediate of farnesyl pyrophosphate. And the condensation of 2 farnesyl pyrophosphate groups generates squalene, which is a 30-carbon intermediate. Both farnesyl pyrophosphate and geranyl pyrophosphate are important intermediates in this process, as these can be used to farnesylate or geranylate proteins. This is a modification similar to phosphorylation of proteins, where it will change the activity of the molecule. From here, our squalene molecule is oxygenated by squalene monooxygenase. This creates a high-energy epoxide ring. An opening of the ring facilitates cyclization into our first cyclic intermediate, linosterol. From there, linosterol can be converted into cholesterol. So once cholesterol is synthesized within the cell, there are several fates that we'll look at. One of the fates of cholesterol is the esterification to a cholesterol ester. And this is where a fatty acyl-CoA group is added to the hydroxyl group. The enzyme that catalyzes this reaction is ACAT, or acetyl-CoA cholesterol acyl transferase. These cholesterol esters are more hydrophobic than free cholesterol, and in the case of the liver, these are packaged in VLDL particles and are transported to the periphery. Hepatic cholesterol also serves as a pool for the synthesis of bile acids, which are required for digestion. If you remember, these are detergents that emulsify our fats. They're introduced into the biliary ducts of the liver and are stored in the gallbladder. For the synthesis of cholesterol to bile salts, the first step in that reaction is the conversion of cholesterol to 7-alpha-hydroxycholesterol. In this step, a hydroxyl group is added to the 7-carbon, and this is done through a series of reductive reactions that requires NADPH. The enzyme that catalyzes this reaction, the 7-alpha hydroxylase, can be inhibited by high levels of bile salts and is, again, our regulatory enzyme for the pathway. From here, the 7-alpha hydroxycholesterol will undergo additional oxidation reactions as well as reduction of the double bond in the B ring of the molecule. This series of reactions generates two primary compounds, chendeoxycholic acid and cholic acid. You can see here the double bond in both of these ring structures has been reduced, and in the case of cholic acid, we've added an additional hydroxyl group on the 12 carbon. You'll notice that these side chains are shown as ionized. That's because the pKa for these molecules is 6. So at a pH of 6, which is our pH in the small intestine, 50% of these molecules will be ionized and 50% will be protonated. This explains the flexible terminology where bile acids and bile salts are sometimes used interchangeably. The bile acid is technically the ionized form of the molecule. So as these products work as emulsifiers and detergents, the ionized form is more soluble and more effective at this process. So to facilitate that, we can conjugate bile acids with other compounds to reduce the pKa of this side chain. Two common modifications to cholic acid are shown here, where the addition of taurine to the side chain or the addition of glycine to the side chain dramatically reduce the pKa. In the case of taurocholic acid, the pKa is 2, and now this is almost entirely ionized at a pH of 6. And the same for glycochoic acid, that has a pKa of approximately 4. Again, the lowered pKa's increase the solubility of these molecules and increase the emulsification of the fats during digestion. So as we've seen during fatty acid and triglyceride digestion, the bile salts used for this process are recycled. Nearly 95% of them are reclaimed in the enterohepatic circulation. The bile salts secreted in the feces are going to be what's referred to as secondary bile salts that are modified by bacteria in the gut. These bacteria will deconjugate the bile acids as well as dehydroxylate them, and this reduces their solubility and makes it difficult for them to be reabsorbed. So our primary bile salts, shown here, the cholic acid and the chendeoxycholic acid, are primarily reabsorbed. The secondary bile salts, where we have the removal of our hydroxide group from the 7 carbon, as well as in the case of the thoic acid from the 12 carbon. And these secondary bile salts are, again, less soluble and are mostly excreted in the feces. The secondary bile salts that are recycled are not rehydroxylated, but they can be reconjugated on the side chain to increase the solubility. Before moving on, let's stop and ask a question. HMG-CoA reductase activity would be reduced under all the following conditions except. What about if SREBP So our sterile response element binding protein is bound to its partner SCAP in the ER. If these two partners are bound in the ER, this is associated with cholesterol, and this transcription of HMG-CoA reductase is reduced. So this would definitely occur. So that makes C easy. Elevated levels of sterols, or cholesterol, facilitate this interaction and keep the activity of HMG-CoA reductase low. HMG-CoA reductase, remember, is also regulated at the level of phosphorylation. And phosphorylation of HMG-CoA reductase by AMP kinase will also reduce the activity of the enzyme. An additional role for elevated sterols, or cholesterol, is the targeting of HMG-CoA reductase for degradation. The only thing that doesn't fit here is B, elevated levels of insulin. Under these conditions, insulin will activate a phosphatase that will dephosphorylate any phosphorylation on the enzyme and increase the activity of HMG-CoA. So again, if cholesterol is bound to our transcription factor complex in the ER, this will reduce HMG-CoA activity. Elevated levels of sterols will promote HMG-CoA reductase for degradation, decreasing the amount of protein, and AMP will phosphorylate the enzyme, reducing its activity. The only thing there that didn't make sense was insulin. Elevated insulins promotes the action of a phosphatase that dephosphorylates and activates the reductase.